Hi, this is Professor Cam, and today we're talking about transcription. And overall, what transcription is it's what turns DNA to RNA, and it consists of 3 steps: initiation, elongation, and termination. The first step is initiation, which is a little different between prokaryotes and eukaryotes. And first, we're going to talk about what happens in prokaryotes. So basically, you have your DNA strand, and you have a promoter, which is right here, and then your RNA polymerase binds to the promoter, which is happening right here, and so that causes this DNA strand to start to unwind. The process is more involved in eukaryotes because the RNA polymerase can't directly bind to the promoter without transcription factors, or the transcription initiation complex is also part of eukaryote transcription, which we'll talk about in a minute. So now we're going to talk about eukaryotic initiation, and which is a little different from the prokaryotic in that you have a nucleotide sequence, which is basically TATA, and we call that the TATA box, which allows transcription factors, which we talked about in the last video, to attach to the promoter so that the whole transcription process can carry through. And so the transcription factors attach to the TATA box, and it keeps adding on to create so that RNA polymerase II can also attach and form the transcription initiation complex, and then this allows for the DNA to eventually unwind and RNA synthesis to begin.
How is RNA transcription regulated? There are specific transcription factors that then follow general transcription factors. Specific transcription factors include control elements. Control elements are non-coding segments of DNA that regulates protein binding, which regulates transcription. Now let's draw a gene. With a poly-A tail and a promoter. There are 2 types of control elements: proximal near the promoter, and we will focus on distal control elements located further upstream, which utilize enhancers. With 1 enhancer per gene, one gene can have multiple enhancers. The rate of gene expression is regulated by the number of activators and repressor proteins binding to the enhancers. These are activators, while they bind to the enhancers. First, the activators attach to the enhancers. Then DNA bends by specific DNA binding proteins. Third, mediator proteins connect to the activators. And finally, usual transcription occurs when general transcription factors come to help. RNA polymerase II attaches to create a transcription initiation complex. In conclusion, the main idea is that activator proteins bind to DNA enhancers to form activation domains that call other regulatory proteins to start transcription. And transcription is all facilitated by protein-protein interactions.
Hello everyone, my name is Professor Salman, and today I will be teaching you an AP Biology lesson about operons. So before I get started, I would like to go over a few definitions to set the stage about what operons do and how they function in both eukaryotic cells and prokaryotic. An operon is basically a unit made up of linked genes that is thought to regulate other genes. each operon contains a regulatory DNA sequence, which is very important. And that DNA sequence acts as a binding site for regulatory proteins, which are also very important to the process of this operon, that promote or inhibit the process of transcription, which is also another key thing. So what regulatory proteins are basically, they're these molecules, and they have small molecules bind to these molecules known as ligands, which can make the protein active or inactive by changing its ability to bind DNA. So it's important to keep in mind that these ligands can either make the protein active or inactive. Very important. So you can have two types of operons. One type of operon is known as inducible operon. And an inducible operon, what it does is it can be turned on by the presence of a smaller molecule, like a ligand, for instance. And you can have operons that are repressible, and these repressible operons, they can be turned off by smaller molecules. Thinking about it in more of an English perspective, repressible meaning kind of like turning off, and induce meaning turning on or starting up. Thank you, guys. Hi, guys. We're back again, and now I will basically talk about where these operons are actually located. So one example is, a general example, is we have this bacterium, for instance, okay? Bacterium. And inside the bacterium, we have this small bacterial chromosome. Bacterial chromosome. And in this bacterial chromosome, a specific section of it is your operon. So now, that specific operon, if we expand the view of it, this is how that operon will look like. So you have basically a bunch of genes in the operon. Let's just say gene 1, gene 2, and gene 3. And some other sections here. And this is basically your DNA sequence. And you have this activator region, this promoter region, and you have an operator region. So basically, this promoter region, what it does is you can have many specific proteins bind to it. And specifically, one protein that binds to this specific promoter region is called RNA polymerase, is what we'll be seeing in specific examples. And your operator region is basically a negative regulatory feedback mechanism in a sense. And what it does is it represses transcription. So if you have a protein bind to usually the operator region, it's basically a repressor, and it will repress transcription. And then you have this activator region. Activate, it's more of this positive feedback in a sense, and it enhances or promotes in a way transcription. And this basic operon we have here, what it goes through, these genes work together, all of these genes here. And they go through the process of transcription. And after they go to the process of transcription, you have an mRNA sequence. mRNA. Gene 1. Gene 2, gene 3. And this mRNA sequence, after the process of translation, all of these genes will turn into proteins. protein 1, protein 2, and protein 3. Basically, this is a basic operon. Hey guys, Professor Salman is back, and today we will be specifically talking about the lac operon. So basically, the lac operon is part of the E. coli genome. Okay, it is part of the E. coli genome, and what this operon does, it codes for genes that help metabolize lactose. So codes for genes that help metabolize lactose. Hence that lac, stands for lactose. And basically what this operon looks like is basically you have, again, this and this. And for emphasis, I'm going to use different colors here to really reinforce the concept. Basically right here is you have your cap site, CAPSIT site. And then over here you have a promoter. Okay, a promoter. And over here you have another region on the operon called the operator region. And then finally you have 3 genes on this operon, which include LAC-Z, LAC-Y, and LAC-A. Okay, so these are your 3 genes. And what these 3 genes function to do is that they help, again, metabolize lactose. So this is your whole operon. And what this cap site is basically you have this cap protein, CAPPROTEIN, cap protein. And this cap protein, basically at specific times, it can bind to the cap site and acts as this positive regulatory mechanism that allows RNA polymerase, which is another protein, to bind to the promoter region. Okay, so you have RNA polymerase. So basically what this cap protein does is it acts as a positive regulatory mechanism, and it binds to the cap site at specific times, and this allows RNA polymerase to bind to the promoter region, which promotes a process known as transcription. And there's also another protein in this process. It's called the lac repressor. And what the lac repressor can do is basically, if it binds to the operator region, it represses the process of transcription. So basically, in this video, we went over the general overview of a lac operon and what it looks like. And in our next videos, we will go over 4 specific scenarios on how each of these proteins will bind specifically to these sites, depending on glucose and lactose levels, and see whether transcription will be promoted or inhibited. So in the previous part of this lac operon explanation, we were talking about looking at 4 scenarios which could happen specifically with the lac operon. So in this specific scenario, which is the first scenario we will be looking at, glucose is present and lactose is absent. So we talked about that the lac operon, the function of this is to code for genes that help metabolize lactose. If you think about it logically though, when lactose is absent, the operon does not need to metabolize lactose. So you do not need transcription to occur, which makes total sense because you do not need to metabolize lactose if you don't even have it in the first place. So now how this process works is, as we talked about, there's this cap site, this promoter region, and this operator region. And this cap protein goes along with this cap site. However, since glucose is present in this situation, which means we have high levels of glucose, increased levels of glucose. That means this ligand molecule, which is a small binding molecule known as CAMP specifically to the cap protein in this specific site, it will not bind, because if there's an increased amount of glucose, then that means there's a decreased amount of CAMP. So if there's a decreased amount of CAMP, then that means this ligand molecule will not bind, X mark, will not bind to the CAP protein. So since it does not bind to the CAP protein, that means this protein is inactive. It cannot bind to the CAP site, and as a result, it does not specifically help RNA polymerase bind to the promoter region. Okay, so that's important. So there we have that. And specifically in this situation, since we do not need to metabolize lactose because we don't have lactose, this repressor protein right here, this specific protein right here, you see specific shape, it will be like a puzzle piece and go into here, and it'll look like this. So you'll have your repressor protein right here. It's specifically in the lac operon. This repressor is called the lac repressor. It will be bind to this operator region. And since it's bound to the operator region, this RNA polymerase will not be able to transcribe and go through the process of transcription. So usually you'll have the process of transcription, right? It's transcription. But in this case, it's a big no. The reason is because you have this lac repressor bound to the operator region. So here we have it. This is one of the scenarios when glucose is present and lactose is absent. Okay guys, we are back here with scenario 2 in the lac operon. So basically in scenario 2, glucose is present and lactose is present. So in this scenario, both glucose and lactose is present. And when both of these things are present, you will have a low level of transcription occurring. And let's explain why that occurs. So basically, again, like I explained in the first video, I don't want to repeat too much. We have this cap site, this promoter site, and this operator site. And we have this CAP protein that goes along with the cap site. But in this situation, since glucose is present, increased glucose, that means we have decreased amounts of CAMP. CAMP is a ligand that binds onto the protein, the CAP protein. And if there are low levels of CAMP, it cannot bind to the CAP protein, which means you won't have the binding of this to the CAP site, which means you will not have the promotion of RNA polymerase binding to the promoter region as much. However, the RNA polymerase molecule still is, to a little extent, going to bind to the promoter region. And in this situation, you have this lac repressor, right? But since lactose is present, an isomer of lactose, this isomer right here of lactose, known as allolactose, which is also written here as well, acts as an inducer in this situation, and it will bind to the lac repressor protein, which basically disjoints the operator region from the repressor. So the repressor is not on the operator region. And since it's not on the operator region, you're going to have transcription occurring, low levels of it. Transcription occurs, and you will have some metabolism of lactose. So you will have some conclusion, some metabolism of lactose. Okay, so basically this is the third scenario we can get with the lac operon. And in this scenario, you have glucose absent and lactose absent. So basically what that means is if glucose is absent, decreased glucose levels means you have an increase of CAMP. Okay? And this increase in CAMP, basically what that does is you have an increase of this ligand molecule, which will allow CAMP right here, this is CAMP, to bind to your cap protein, CAP protein, and that makes this protein active. And this active protein will bind to this site with your thing. So CAP protein will bind with the CAMP molecule, and it will be in the cap site. So once it's on the cap site, what it does is it signals RNA polymerase to come and bind to the promoter region, which you can see here, and you have the binding of RNA polymerase faster and quicker. However, since lactose is absent, you have your lac repressor bound to the operator region. You have this repressor bound to the operator region. So you see this repressor here? It will be bound. Since there is no lactose, you do not need the metabolism of lactose to occur, which is the job of the lac operon. So you have this repressor right here bound to this, which means in general, you will have this will be the transcription that will be occurring. However, since this repressor is bound, that equals no transcription. No transcription. Big no. So basically, the conclusion in this situation is you have no transcription occurring. Okay, so basically this is the fourth and last scenario with the lac operon. In this specific scenario, glucose is absent and lactose is present. So let's address the glucose being absent first. So decreased amount of glucose means an increased amount of your CAMP, okay, cyclic AMP. And this is your ligand molecule in this situation, which is just a molecule that binds to your protein. So this is your protein binding site, and your CAMP comes in and it binds to the CAP protein. This activates the CAP protein, and what that allows to do is the CAP protein can come and join to the CAP site. And basically what this does is the binding of the CAP protein to the CAP site, it allows RNA polymerase to quickly, more fastly, and more efficiently, which is located here, bind to this region called the promoter region. Okay, so that's basically addresses the glucose being absent. And then the lactose being present, what that does is, since you have lactose, that lactose turns into the isomer, which is structurally the same, structurally different, but chemically the same. It's called allolactose. And this specific allolactose acts as an inducer in this situation. So what it does is it binds to the repressor protein, And since it binds to the repressor protein, your allolactose right here, it goes off of the operator region. So this is the operator region. And since allolactose is bound to your lac repressor protein, it's not going to be in the operator region. So two factors here. Basically, you have your CAP protein bound to your CAP site, and you have your RNA polymerase bound to your promoter region, and your repressor, your lac repressor, is off of the operator region. So this basically stops repression. And since it stops repression, what you have is a beautiful and harmonious transcription occurring. Very strong transcription. So that's what we have to get out of here. That's the conclusion. Conclusion is that we have very strong transcription occurring in this situation. And it makes sense because if you don't have glucose and you have lactose present, then you need to break down this lactose in order to get energy. And the best way to do this is allow transcription to occur and have these genes, all of your lac genes, metabolize lactose